Hello, my name is Eunice Lim, and this is my video lecture for Molecular Biology in Conservation course. It is titled Genetic Markers. I will specifically be talking about chloroplast genes. The chloroplast is theorized to be formed through endosymbiosis. The endosymbiotic theory is that hundreds of millions of years ago, a large prokaryote cell engulfed a cyanobacterium, which remain intact within the large prokaryote and begin to live as symbionts. The cyanobacterium could perform photosynthesis. This is believed to be the ancestor autotrophic eukaryote. These ancient eukaryotes had an advantage in their endosymbiosis. The prokaryote evolved to become chloroplasts. This is proven as chloroplasts have their own DNA, which are arranged similarly to prokaryote DNA, specifically bacterial DNA. The most important function of the chloroplast is to synthesize food by the process of photosynthesis. It absorbs light energy and converts it into chemical energy. Chloroplasts have a structure called chlorophyll, which function by trapping the solar energy and use for the synthesis of food in all green plants. Chloroplast produces NADPH and molecular oxygen by photolysis of water. The chloroplast also produces ATP by the process of photosynthesis. The carbon dioxide obtained from the air is used to generate carbon and sugar during the Calvin cycle or dark reaction of photosynthesis. Now I will share with you the structure of chloroplasts. Chloroplasts are found in plant and algal cells. In flowering plants, chloroplasts are normally found in the mesophyll cells of leaves. A typical cell has about 30 to 40 chloroplasts. Many chloroplasts are biconvex discs, 3 to 10 micrometer long and 2 to 4 micrometer wide. The chloroplast envelope consists of two membranes, a more permeable outer membrane and a selectively permeable inner membrane. A system of interconnecting membranous flattened sacs are called thylakoids. The thylakoid membranes are arranged in stacks called grana. There are about 50 grana in a chloroplast, and each granum is made up of between 2 to 100 thylakoids. The thylakoid membranes are surrounded by a semi-fluid stroma. Stroma contains other elements such as circular DNA, starch granules, and lipid globules. Two commonly used genes from the chloroplast are RBCL gene, which codes for the large subunit of rubisco, an enzyme involved in the first major step of carbon fixation, a process by which carbon dioxide is converted to glucose by photosynthetic plants and organisms. The RBCL gene has been commonly used as a suitable locus for analysis of phylogenetics in plant taxonomy. The next gene is the MATK gene, which codes for matuase, a protein that splices group 2 introns. Many RNA transcripts require MATK for intron excision. These transcripts include tRNA, tRNK, tRNL, RPS12, RPL2, and ATPF. The tRNA or protein products from these genes are required for chloroplast function, including photosynthesis. The two more rare genes that is used in molecular biology is SAG genes, which are involved in the senescence of plants. The senescence of plants is the sequence of degradative processes leading to the remobilization of nutrients and eventual death of the plant. INFA is also another rare gene that is used. It is important for the initiation of protein synthesis, ribosome binding, rRNA binding, and translation initiation. All of these 4 genes, the RBCL gene, the MATK gene, SHG gene, INFA are protein coding genes. So why are the two genes more commonly used rather than the other two genes? So the RBCL gene is an abundant protein in leaf tissue of plants, and it has high PCR amplification success, whereas for MUTK gene, it has high evolutionary rate. These two genes, both the RBCL and MUTK genes, are considered standard plant DNA barcoding markers because they are universal. They have relatively high overall sequence quality. They are low cost and high discriminatory power. So that concludes my video.